Okay, so in today's video, we are going to look at some of the processes that occur in a cell. Okay, so first we're going to start with the membrane and some of the processes that the cell membrane is involved in. So really what the membrane is going to do is it is going to prevent or allow the movement of substances into or out of the cell. So there are two categories of processes that we'll be talking about, and they are passive and active transport. So really the difference is the requirement of energy. So passive transport does not involve any energy. So it just happens. And active transport is going to require an input of energy, such as from the breakdown of ATP. One type of passive transport, so requiring no energy, is diffusion, which I'm sure that you are knowledgeable on at this point. So diffusion is just the movement of particles from high to low concentrations. So if on one side we have a lot of something and on the other side we have not a whole lot, then the movement will be from where there's a lot to where there's a little. Okay, think about like if you were in a room and there's a whole lot of people on one side, you probably don't necessarily want to be around all those people. So you would kind of want to move away so that you would have a little bit more room for yourself. So that's the same idea here, is that through the membrane, particles are going to run from high to low concentrations, basically down the concentration gradient. Okay, so the concentration gradient is just the difference in concentrations on either side of the membrane. And then the movement in diffusion is going from high to low. Okay, so here we can see some dye being put into some water. So it's going to be very concentrated in the area where the dye goes in, but over time, you'll see that the color is going to spread out until it's uniform. Okay, so this whole flask of water would be pink rather than like what you see here. All right, so over time, they would move from the side of high concentration to the side of low concentration until it's even. Okay, and then there would still be movement back and forth, but there would be no overall change. Our next one is facilitated diffusion. So with diffusion, okay, if you think about it, it's passing through from, just right through the cell membrane, then these particles are nonpolar. So facilitated diffusion is going to be a process that allows polar molecules or larger things to pass through the membrane where they normally would not be able to because of the polarity, the non-polarity of the phospholipids. Okay, these things, they'll just bounce right back, right? But there are proteins in the membrane that will allow, kind of like a little tunnel way for these particles to pass through. Okay, so some examples of things that use these transport proteins are ions, which are charged, glucose, which is polar, and amino acids, which can be polar or charged even. Okay, but just like with regular diffusion, facilitated diffusion will also go down the concentration gradient. So this process is very important for muscle and nerve function, which we'll get to later in this year. Okay, but basically ions are super important and used for, electrical signals. So they'll go from a large concentration on the outside or the inside to the other side, and that will alter the membrane's potential. Finally, we have osmosis, which is simply the diffusion of water. So water works a little bit differently, okay, in some ways, because water is going to move from the side of low solute concentration to higher solute concentration. So really what we have here is on one side, we have a larger proportion of water to solute than we do on the other side. So the water is going to move to the other side so that it balances out that concentration. Okay, so the solute concentration is lower on the side that it is moving from than the side where it is moving to. But if you think about it in terms of just the water, there would be a larger percentage of water on this side than on this side in relation to solute. Right, so we have three different types of solutions which will determine the movement of water. If it is isotonic, then that means that the concentration of solute is the same on both sides of the membrane. So there will be no overall movement of water. If it's hypotonic, then that means that there will be a lower concentration of solute on the outside, which will mean that the water will flow inside of the cell. It will go into the cell, which will make it swell up. Okay. Hypertonic is going to be the opposite. So there's a higher concentration of solute on the outside of the cell. So that means that the osmotic pressure is higher on the outside of the cell. So that will cause water to flow out of the cell, which will cause it to sink because it won't have as much water in it as it did before. So it'll kind of look shriveled. All right, so here's a nice little diagram here. So if it's a hypertonic solution, that means that out here there is higher concentration. So water will want to balance that out so it flows out here so that the solute concentration in here will be about the same as what we have out here. And that causes the movement of water out, which will make the cell shriveled. Hypertonic will be the opposite. So there's a higher concentration here, so water is going to flow in, which will cause the cell to swell up like this, and it could actually cause it to burst. Isotonic is nice; it's balanced on either side, so there will still be movement into and out of the cell, but over time there will be no difference in concentrations. Okay, nothing is going to change overall over time. Okay, so some few other processes. Filtration is when molecules are forced through membranes. So if you use a filter, like a coffee filter, the coffee filter is holding back the coffee grounds, but as water is percolated down onto the coffee grounds, the gravity is actually going to pull the water, okay, through the coffee filter and down into the coffee pots. So an example within the body is blood pressure. So blood pressure is really, really important for pushing water outside of the blood vessel in order to get it out, okay, because otherwise water would not move freely through the membrane very easily. Okay, so blood pressure helps to kind of force it as well as some other substances out of the blood vessel. Okay, and that is, blood pressure is helped by having large proteins on the outside, which will make the water want to go on the outside because having that high concentration on the inside of the blood vessel. Okay. So, a little bit more information here. So osmosis will normally prevent too much fluid within the tissue. Okay, so it will prevent really too much forced water out of the blood vessels. Okay. But there are certain conditions where excess tissue will accumulate, right? Excess tissue fluid will accumulate because it's being pushed out. So that is a condition called edema. So when you get injured, whether it's something like a cut or an infection, this will happen naturally to help to heal by sending more fluid out to that tissue to help to heal. Okay. Yeah, we have not done the strawberry DNA lab, but basically what happened in the strawberry DNA lab is that the water holding the DNA was forced through the filter paper through gravity, which is kind of what I just explained with the coffee. All right, now we get to active transport. So this is going to require an input of energy from ATP. So we've got endocytosis, pinocytosis, phagocytosis, and receptor-mediated endocytosis. So endocytosis is just, the taking in of something. So it includes all of the other three. So basically, the membrane is going to surround whatever it is that the cell wants to take in, and then it will form a vesicle around that and basically bring that into the cell. So the membrane surrounds it and pinches off and creates that vesicle that will enter the cell. So this is typically large molecules, or in this case, it could be actually fluid that the cell wants to take in. So the membrane engulfs the molecule and pitches off into the cell. First, we have pinocytosis, which is just considered cell drinking. So this is going to involve the intake of liquid into the cell. Phagocytosis is for larger molecules. It's called cell eating. Okay, so again, taking it into a vesicle, and one major example of this is actually with phagocytes, white blood cells that will attack bacteria or larger cell debris will actually engulf the whole thing, bring it in, and then destroy it, break it down into smaller parts so that the cell can actually use those molecules to build new things. Receptor-mediated endocytosis is for specific things. So there are receptors on the outside of the membrane that will bind specifically to a type of molecule. So when they do, that the vesicle will then pinch off and bring those specific things into the cell again within a vesicle. Okay, so an example of this would be cholesterol being brought into the cell. Right, so the opposite of endocytosis is exocytosis. And exocytosis, again, is going to involve the use of energy, but now things are being sent out of the cell. So there are vesicles being made within the cell at the Golgi apparatus, and those vesicles sometimes will just go within the cell, but a lot of times will actually go to the membrane and deposit these things outside of the cell. So essentially the vesicle will come, bind to the membrane of the cell, and then dump the stuff outside. Okay, so the secretion of proteins, but also nerve cells releasing neurotransmitters like acetylcholine too. So that brings us to mitosis. And we're not going to go into too much detail here in mitosis, but this is the whole cell life cycle here. And we start with interphase, which actually is not shown in this diagram. Or no, I'm sorry, it is. So we have interphase up here, which is the majority of the cell's life cycle, the time spent in it. So here we have gap one, which is when the cell is growing, building more macromolecules, building more organelles. And then we get to synthesis, which is where DNA replication will happen, which is needed whenever the cell is about to go into mitosis. So now we have two full sets of DNA, two identical sets of DNA, And then we get to gap 2, which is where more growth is going to happen. And then we finally get to mitosis, which is where the cell will split and make 2 identical copies. And now cytokinesis is where the 2 cells will break apart. And now both of them will start again at gap 1. Oh, there you go. I don't know why it took so long for those to come up. But yeah, so we have gap 1, synthesis, gap 2. So growth, DNA replication, more growth, as well as organelle replication. And then we get to prophase, metaphase, anaphase, telophase before finally cytokinesis and starting the whole life cycle all over again. Okay, so, here basically same descriptions as I just gave you. All right, I really like this diagram though. I think it's really nice and helpful. A little bit more detail about prophase, metaphase, anaphase, and telophase. So prophase is where the chromatin, which is kind of unwound DNA, will condense into chromosomes. So essentially it's like, you're preparing for a vacation, so you pack all your things into a suitcase rather than just kind of having them all in a ball and carrying it with you. Suitcase will make it a lot easier. Same idea here. So the chromatin is being condensed into chromosomes so that they can be transported more easily. The nuclear membrane will disappear, so that will release the chromatin as chromosomes out into the cell. And then centrioles, which you'll remember is one of our organelles, will migrate to the poles of the cell. That brings us to metaphase, which is where the centrioles will send out spindles, which will attach to the chromosomes and will help to line up the chromosomes along the center of the cell, along what is called the metaphase plate. Then we get to anaphase, which is where the chromosomes split. All right, they're being pulled by the spindles and they split and they will be taken to opposite ends of the cell so that on one side we have one copy of the DNA and then on the other side we have the exact same copy of DNA. And then with telophase, the spindles, which I said attached to the chromosomes, will disassemble and just go away. Chromosomes will unwind and go back to chromatin, which is the form that will be able to be used to, for protein synthesis. And then finally, the nuclear membrane will come back, all right, on both sides. There you go. So prophase, you'll just basically see, you'll go from having DNA being like really long and squiggly to being much more condensed, to having them lined up, to having them pulled to both sides. So you have 2 groups of DNA. And then finally with telophase, they start to unwind again and you have 2 very distinct nuclei in the same cell that are being formed before they finally pinches off. All right, so the very last thing that we have for today is talking about tumors and cancer. So this is essentially uncontrolled mitosis happening. So you can have 2 different types of tumors. They can be benign or malignant. And benign is going to be non-cancerous. It's not moving, right? It's not affecting nearby tissues. It's just right where it's at. It could be possibly having an effect where it is, but it's not moving into anything else at all. So a benign tumor is much less dangerous than a malignant one, which has the ability to move and basically extend into other living tissues and cause problems around there. Okay, so a process called metastasis is where the cancer will spread to other parts of the body. So like, for example, with lung cancer, okay, that would be affecting just the lungs, but if it metastasizes, then it will spread into other parts and affect other parts and cause cancer there. All right. So there are a couple different genes that will contribute to the formation of cancer. And they're called oncogenes and tumor suppressor genes. So oncogenes, when they're activated, will increase the cell division rate. Okay, so that has its uses, but if these get activated because of a mutation or something like that, then that can cause an uncontrolled cell division rate. Tumor suppressor genes help to hold mitosis in check. So these kind of work against each other. But if this gets turned off and this gets turned on, just think about what would be happening. The mitosis would just be happening over and over and over again. And the normal checks that would prevent that from happening would not be there. So these cells could be heavily damaged or mutated, but they're still undergoing mitosis. And essentially, if they're really, really mutated and damaged, then they're not going to be doing their normal job. And that's the main problem with cancer. Okay. So that finally brings us to talking about stem cells, which have the ability to produce specialized cells. So they are not specialized, but they will undergo mitosis, and those cells can then specialize, into a certain cell. So a progenitor cell is a cell that will have come from a stem cell that is beginning to become specialized. So it will undergo mitosis and start producing cells that are specialized and will have a specific use. A totipotent stem cell has the ability to make every kind of cell. Right? So it's just a cell that has the genetic information and has the ability to produce new cells that can become anything. A pluripotent stem cell is a cell that will give rise to specific types of cells. So it's becoming to start. It is starting to become specialized at that point. Okay, so that brings us to the end of chapter 3 with all these cell processes. So I hope that this was useful. But yeah, go ahead and answer the questions in the Google form. And that will be all for today.